My question today is whether the B cells that emerge from the bone marrow into the periphery are functionally mature. And the answer to this question is that they are not yet mature. They will need to arrive in the spleen to become mature. So the bone marrow generates B cells which are still functionally immature. These will have to move to the spleen to become immature. We tend to learn that B cells are produced and mature in the bone marrow, hence B cells, whereas the T cells are produced in the bone marrow, but they travel to the thymus to mature. No. The B cells are produced in the bone marrow, and through the blood circulation, they will travel to the spleen to mature. Now, the thing is that most immature B cells that leave the bone marrow will not eventually survive to become fully mature B cells. The daily output of new B cells from the bone marrow, so the output, is about 5 to 10% of the total B lymphocyte population in the steady state peripheral pool. The peripheral pool now of the B cell population is the spleen plus the lymph nodes. This is what we call the peripheral pool. So if there's an input of 5 to 10% of new B cells in the peripheral pool, and we now know that the size of this pool seems to remain constant due to homeostasis, we would expect, now this is the important thing, that the stream of new B cells needs to be balanced by the removal of an equal number of peripheral B cells. So you would expect peripheral B cells to die which are mature at the same percentage. However, the majority of peripheral mature B cells are long-lived and only 1 or 2% of these die each day. So 1 to 2% die each day. So if the input is 5 to 10, the output is 1 to 2%, the peripheral pool shouldn't be constant. The population shouldn't be at a steady state. What actually happens is that most of the B cells that die are in the rapidly turning over immature peripheral B cell population. So more than 50% of this immature B cell population, the output, in other words, of the bone marrow, will die every 3 days. Now, why does that happen? Why more than 50% die every 3 days? The failure of most newly formed B cells to survive for more than a few days in the periphery seems to be due to the competition between the peripheral B cells for access to the follicles of the spleen. if newly produced immature B cells do not enter a follicle, the passage through the periphery is halted and they eventually die. So if they enter the follicle, they will leave and mature. If not, they remain outside and they die. Why is that? Because inside the follicles there's a B cell activating factor that will allow them to survive and mature. So inside the follicles now, inside the lymphoid follicles, we've got a TNF family member, which is called BAFF BAF. It's the B cell activating factor. This is produced by the follicular dendritic cells, which are found inside the bone marrow, inside the follicles of the spleen, FDCs. These have nothing to do with the dendritic cells that you already know. They are not hemopoietic cells. They reside in the B cell follicles and they are specialized to capture antigens for recognition by B cell antigen receptors. So nothing to do with the dendritic cells that you know. Now these FDCs express these cell activating factor BAF, and the immature B cells express a receptor called BAF obviously receptor. Binding of BAF to the receptor will allow B cells to mature and survive, of course. Let's go back to these two panels now and try to explain what we've already said. Now, in the first panel, we are in the blood. Now we are entering the spleen. These are parts of the spleen, the marginal zone and the follicle, the lymphoid follicle, which is called B cell follicle. Inside the B cell follicle, you see two types of cells. One of them is the B cell in a transitional state, still immature, and you also see the follicular dendritic cells. Look on the cell membrane of the FDCs, they express this B cell activating factor. On the other side, the B cells also produce and express on the cell membrane the receptor for this B cell activating factor. Binding to this receptor would lead to B cell survival. Newly emigrated transitional B cells at the T1 phase that you see here, T1 phase, exhibit high levels of which other protein? The immunoglobulin M, IgM, and little IgD, and of course the BAFF-R. In the B cell follicles, these cells upregulate also CD21 to become transitional stage 2 B cells. Don't forget that these have now entered already the lymphoid follicles in the spleen, so they will survive and they will mature. So they are at this stage and they will start also expressing CD21. Finally, the cells upregulate also IgD and become long-lived mature B cells. So this is now a mature, long-lived B cell. The majority of long-lived B cells are recirculating B cells known as follicular B cells. Now there's also another subset, the ones that you see here, the marginal zone B cells. Now, the marginal zone B cells are thought to be weakly self-reactive and express very high levels of the complement receptor CD21, the blue one that you see on this image. These cells migrate to the marginal zones of the splenic white pulp part area of the spleen, an area with white pulp, red pulp junctions. In this location, the marginal zone B cells are poised to make rapid responses to blood-borne antigens or pathogens. The transitional T1 B cells that are excluded from the follicles, these ones, which fail to enter the follicle, fail to receive maturation. And why is that? Because they fail to receive survival signals, the BAF signals from the follicular dendritic cells, which are found inside the follicles. And because of this failure without the BAFs, they will die within 2 to 3 days of leaving the bone marrow. So they left the bone marrow, they came to the spleen, they failed to enter the follicles, so they will not receive the B cell activating factors from the follicular dendritic cells, and they will finally die.